All right, so this morning we heard a lot about using blogs in science, but not necessarily using them as the primary vehicle for communicating science, and that's what this session is going to be about. So I'm using the term here open source science. There's quite a bit of history with that term. With the open source movement with software, there's some confusion that actually we're talking about open source software in science, which we're not. We're talking about using data as the actual source for doing the science. So the term that I've been using lately is open notebook science, which is what we do in my lab, where the wiki is actually the official notebook of our lab. And I'll show you what that actually looks like in a little bit. Okay, in terms of an agenda, I posted this a couple of days ago. Again, this session is really only about the dissemination of primary scientific information via blogs, wikis, and you'll see a couple of additional non-traditional vehicles. So in this case, we're not blogging about an article. The blog is actually the primary source of information. Now, the types of information that have been disseminated: raw experimental data, which is something that I'll show you from our useful chem system, but also just the analysis. So you can have people that are not necessarily showing the raw data, but they are talking about the analysis. You can have other people that are not even talking about the data directly, but they're talking about hypotheses, what they think might work. They're writing a proposal. They might want to get some feedback. You'll see so-called failed experiments, and hopefully by the time we're through, I'll convince you that there really is no such thing. There's either properly executed experiments or not. But in fact, the information that you get from a chemistry experiment can always be used if it's recorded properly. I'll show you examples of generalized protocols. So these would not be a specific experiment necessarily. These would be general ways of doing things, and that's something that people are using blogs and wikis for. And the traditional article format. I think we saw an example of that this morning with the circadian rhythm, where there was an introduction, conclusion, and all that stuff. So that can also be done. I want to go over a couple of issues, and this is where I'm asking you to think as I'm talking. If you perceive some additional issues, I'd love to incorporate them in this on the wiki for this page. Intellectual property is one that always comes up. The way that we handle it is we just avoid it. We're not interested in patenting what we're doing, so we're just donating it to the public domain immediately, and it actually says that at the top of the wiki and the blog. If you do want to retain intellectual property, in the US you may be able to within a year after you publish it, but it gets pretty tricky, and I wouldn't recommend doing open source science if you were really interested in maintaining that. There's the issue of referencing and claims to priority. One thing I want to show you is why a wiki particularly is so good at referenceability and actually being able to say that you did something at a certain time. There's the issue of academic validation. So we're talking here about tenure and promotion. How is this kind of scholarship going to be evaluated? And again, that's a question that was started this morning. And we didn't hear that much actually about the peer review process, but that's something that will come up when we are publishing our raw data. Blogs and wikis, as you all know, do have a form of peer review, although it is more elective. If no one wants to comment on your post, they won't. So there are systems that people might want to create to make the peer review mandatory in certain ways, and there's nothing that I'm doing that prevents that. It's just that we're not doing that specifically. Okay, the third thing, so that was a little bit of the negatives. Here's a little bit more of the positive side, the opportunities that we have from this. Increasing productivity in terms of universally usable knowledge units. The one thing that I'm going to try to get across is that the research article format is not necessarily the best way to communicate scientific information. It's good when you have a story with beginning, a middle, and an end, and you want to use arguments. It's sort of like a court case, right? You want to support your point, and anything that doesn't support your point, you can't really put it into the paper. But we all know that knowledge is not like that, right? Scientific research generates stuff that cannot be classified into neat buckets. So using a blog or wiki to disseminate your scientific results actually will lose nothing. Everything will get captured. The second issue is making explicit the nature and quantity of work and collaborations. And again, especially with the wiki here, the contribution of everybody can be broken down to their posts, right? And in a wiki, it's their version, and you can see what they added, what they deleted, and that becomes extremely clear. So if you wanted to evaluate somebody, you wanted to hire someone, and they were recording what they were doing on a wiki, you could get an extremely detailed analysis of their knowledge base, which is something that is very difficult to tell when you're just a co-author. And the third issue, probably the one that motivates me the most to get involved with this, is if we start to use semantically rich formats, which I'll show you in chemistry, things like CML, chemical markup language, which is just a form of XML, we can start to really seriously think about automation. And if we factor into that zero publication cost, then we may actually have a way to the technological singularity, which is a term that basically means the progress is advancing so quickly that we can't even observe it anymore. And that, I think, is going to happen sooner or later as computational power becomes more and more powerful. The question is just how are we going to get to that point? I think it's going to happen through the social software because it is a bridge between humans and automated systems. And speaking about that last point, so we look at the way science has evolved. Of course, these are supposed to represent people here. So you have two human beings that are exchanging information and they're doing science. And that is, what we have observed for, the past several hundred years. We are, I think, right here, where you do have human beings that are still doing experiments, but we have a lot of automation. We have machines that are doing a lot of the experiments. We have computers that are crunching the numbers, that are analyzing the text. Although right now it's not very semantically rich, so it's not, at a level where it's going to be, but we are reaching that point. At some point, I think human beings are going to become the slowest part of the process, and once that last step is removed, it'll be very interesting to see what happens to science. When machines are designing experiments, executing them, and based on the feedback, redesigning new experiments. That's when, things are going to get very interesting, I think. So we see some evidence of this. There are people who are pushing in this direction. This is the robot scientist. Ross King is the one who created this. And this is a machine that is capable of creating hypotheses and evaluating them and doing experiments in yeast. So it's a controlled experimental space. In other words, the machine isn't going to do like Diels-Alder reaction, but within that space, it can grow yeast, it can analyze its metabolites, it can do all of these things and be in a closed system and actually discover new knowledge. So we want to move towards a situation where we're doing something like that using organic chemistry and not just genomics. Okay, so how can this happen? Well, again, if you have self-organizing redundant processes, you have agents that participate with zero cost. One of the issues with the publication models that we've looked at so far, either the readers are paying or the authors are paying. I don't know. I mean, the life scientists have it better, I think. But as a chemist, I can't afford $1,500 for every article that I want to make open access. So there has to be another model. And using blogs and wikis, you have a zero-cost system, especially if you use a hosted system. Like I'll show you with Wikispaces, it's actually free and hosted. And again, if you have fully open access, see the point is that all of these things can only happen if everything's open. If there's a database with a toll, actually even if it's just a registration system, it creates a block in the system. But once everything is absolutely completely open, then anyone in the world can analyze my data and can suggest additional experiments. And so that's why, again, I see the wikis and blogs to be particularly useful. Okay. So if we do have machines eventually doing science, how will they know what to work on? And that's how this project basically started. This Useful Chem project started by actually starting with no assumptions and asking humans what is important. And the way that this was done is by using Google Scholar and Cyrus and searching for these terms. What is needed now, what is missing, there is a pressing need for, and seeing what it actually came up with. So this was started in July 2005. And it came up with these things, like there's a pressing need for the identification of more effective dyes, fuel cells. But one that kept coming up is developing new drug-based anti-malarial therapies. And so that one is the one that we ended up pursuing. We're organic chemists, so we had to pick a project that we could actually execute. And furthermore, I found this not-for-profit organization, Find a Drug, that had actually screened a lot of compounds against, I think, like 250 different enzymes for diseases. And I contacted them, and they sent me a couple of libraries. One of them was for enoyl reductase that, if you inhibit it, has a good chance of preventing the replication of malaria. Okay, so these things were coming together. Again, all of this, zero cost. And we don't have a lot of chemists in the audience, so I'll skip through this pretty quickly. But basically, going through the blog, you can see the evolution of the synthetic design. Okay, so explain how to do certain things, and then as I kept reading, discovered that there were better ways of doing it, and we came up with a synthesis. And now this is where the information naturally got parsed. So this wasn't done from a top-down approach. Basically, we created whatever vehicle we needed to distribute the information in a suitable format. And in chemistry, you have molecules. So it made sense to put each one of the molecules of interest, which could be either the molecule we're trying to make, it could be a starting material, an intermediate, basically anything that we were going to touch, to make actually a blog post of it. And we have a picture, obviously, and then we have different ways of recognizing it. So the SMILES code is a way of representing a molecule with just using text, and that's something that a lot of databases use. These are links to commercial suppliers, so Aldrich, and then NMR is a way of analyzing the compounds. And the first thing that seemed obvious to me is to, use a blog as our notebook, right? Because you could have one post per experiment. And that actually didn't work out too well when the group actually interacted with the system. You could, in fact, put a scheme what you were doing, put an objective, all of that. But what happened is that because I had multiple students, with a blog you can't tell what got changed. So if there was something to correct, I could write a comment and say, this is wrong. And then the student would come in and change it, but what they had done had vanished. Okay? And another problem with the blog is that it's difficult, even with a third-party blog, to know the exact date. Blogger, for example, will let you change a date. You can backdate things very easily. And so that could be a problem if you're talking about priority. Okay, but it still provided valuable feedback. Here's one experiment where Matt Todd, this is a researcher in Australia, and he basically was, looking at our experiments and saying that we had a problem. He had a problem with our concentration. So he was giving us feedback on an experiment that hadn't even been completed yet. It was still in progress. But we discovered that it was far better to use a wiki, and I'll go through a number of reasons here. For one thing, with a wiki, you can actually, so we're still using blogs, right? We're still using like the molecules blog, but we are using the wiki as a way of coordinating all that information. So here, for example, is the progress of the things that I told you about. We did a search, we got a library, all of these things. So in the wiki, I can list and link to them. So it's a way basically of people being able to get updated on what we're doing. So I can do things like tell a story about failures that we had. Here's a compound that we tried to make, Dopal. We eventually did make it successfully. And here, experiment 25 is the one if you want to only know the successful way to do it. But there's also a whole thing here about the explanation of our failures, but not only that, why we failed. A couple of them are very interesting because the information in the peer-reviewed journals was inconsistent and some of it was just simply incorrect. And we didn't know that, so we were relying on certain informations that later on turned out to be incorrect. And all of this is available to be looked at. Normally this would not be published, of course. Now the real breakthrough came when we actually moved everything over to the wiki in terms of the experiments, in terms of the recorded experiments. So here we can have your normal. This should look a lot more like a lab notebook page. You have your little scheme, your objective, and these are hyperlinked to molecules in the blog. Okay, so if you click on these, you'll get more information on all those molecules. The wiki lets you see the experiment history. So if I'm looking at experiment 25, I can see the dates, the people, and then I can look at the actual versions of what was done at that time. So one of the ways that I've used the wiki with my students is, instead of just going in there and correcting mistakes that I see, is I would write a question. All right, so here, instead of actually calculating the yield, I basically just put in, you forgot to put the number of millimoles and the yield. And so I would do that, and that's actually been a really good way to train students on how to do science, how to actually interpret results and what's meaningful and what's not. The nice thing about this too, because it's completely open, basically anyone in the world can mentor my students. People can have different background, they can have different expertise. I have my own expertise of what I know in organic chemistry, but clearly you can't know everything, so it's very nice if you could have an expert in different areas. And I'd really like to be able to do that with some faculty around the world. Swap students, basically. We can make comments, and I think that would improve the student population. But the thing that's very nice about the wikis, and again, especially wikis that are hosted by third parties, I use Wikispaces for a number of reasons. I like the interface. I like the way that you have nice RSS feeds for it. It's all pretty good. But the other thing is that I cannot forge this date. This is on the Wikispaces server, and the version date remains forever. So if I wanted to prove that I had done or known something at a certain time, I can easily do that by providing a link to the specific version. And that's what I really like about the wiki.
Do you make and keep local backups of everything on there?
Yeah, Wikispaces, you can actually back it up as a zip file. Now, you can store files. I just use our server. So theoretically, you could go and alter the file in the server. But look, realistically, if you have an experiment and you're talking about it and you're talking about results, I think that's sufficient to prove that you've done something. You can always lie, but this makes it really, really hard.
What are you backing up? Are you backing up the software? Are you backing up database tables?
And Wikispaces, it just comes up, it's a bunch of pages. That's all a wiki is, after all. It's just a bunch of HTML pages.
I guess what I'm wondering is, say you back it up and all of a sudden Wikispaces goes out of business. Do you have the software to...
Well, Wikispace is owned by Google. So I have more confidence in Google than most academic institutions.
But it's HTML pages, right?
Yeah.
The worst case scenario is you just have to transcribe it in a network.
You should always use redundancy regardless of what you do. Yeah.
Well, as long as HTML's
I mean, wouldn't it be safer if the whole thing were hosted on your computer rather than some other external?
No, the question is, would I would definitely trust Google more than a server run by any single individual in an institution. Absolutely, they have to stay in business. They're not looking for the next job. But regardless, that's not what's important. The key thing, it's not so much this one place where everything is. It's the knowledge is being distributed. Here's one place where it is. I'm also blogging about this in another blog. So it's basically it's distributed. So if one piece of it falls out, it's still the information is going to make it to the people who need to have it. So that's very different from the concept of I will publish an article here, and I may never republish that article. So it's a different concept. Okay. So using the wiki also makes it very easy to tell what's going on in our lab. Like right now, if you were to click on the most recent changes, you would see today what my students did. And it just shows up like this. It just gives you the experiment number, when, and you would just click on it and it would take you to that experiment to see what's going on. Okay, so the other thing that I use is SiteMeter, which is, again, everything I try to do with free hosted services, for the main reason that I want anyone in the world to be able to replicate what I do in an instant. So if you like any aspect of this, you can go home and you can replicate this for free. SiteMeter is just a service that will see who is visiting, how many people are visiting you, and where they're coming from, what kind of keywords they're using. And I put it at the bottom of all my wikis and blogs. And that way I can tell what is making it, what's going through. So, I can see other blogs that people are coming through, but I can also see searches like here, chemistry of protease inhibitors. I can see what people are searching for and what my blog is on top of relative to other blogs. And that's something you normally have no feedback in a typical article. You don't even know if people have read it. So another issue here is automation. I'm not going to talk too much about that here, just to give you a little sample. There's a way of representing molecules that's called an INCHI. It's just a code that tells a computer program what molecule you happen to be talking about. The advantage of it is that it's unique. So a molecule has one specific INCHI code, and Google happens to index them very nicely. So if we actually put an INCHI as a tag, it's then very easy, just by going on Google, seeing all the instances of that INCHI coming up, we can track molecules and reactions. Yes?
Is software available to input the INCHI string and get back, a regular molecular representation?
Yeah, the question is there a conversion from INCHI to normal representation? Yeah, you can use ChemSketch, which is a free program. ChemSketch will read smiles and INCHI, and it will also export smiles and INCHI. Yep. Now the automation here too, if you remember that molecules blog, right, which all it was is just, basically it has a smiles code, all right? The smiles code is kind of like an INCHI, it's just that it's not unique. We have a script that runs every day that reads the entire blog and then converts that smiles code into different kind of information like molecular weight. It actually calculates the INCHI and puts it here. And it also goes on e-molecules, which used to be Schmoogle, and it queries it for commercial sources, and then it reports back here. So what that effectively means is that from the human side, all you have to do is to put a smiles code, which is easy to do from ChemSketch, and automatically all of these information fields get populated by a bot. And that's really where we want to move forward with respect to doing science. We want more and more of this stuff to be done automatically. And for those of you who are into CML, we actually create a CML RSS feed. It's like an RSS feed, except it's intelligent. It knows about the molecules, not just about the pictures. Okay, so what's been really interesting about this whole project is, we're working on malaria just based on the simple fact that that's what people were saying in 2005 was a worthwhile thing to do. But we're not the only ones. The Synaptic Leap is another open source biomedical research group. They don't do research, but they have software that they want to coordinate research, and they also have selected malaria. It turns out that it's a really good disease to do open source on because most of the people who are sick can't really afford the treatment, so it's not going to be a huge commercial target, and there's a lot of people that are affected, so it's still a big problem. And so what's happening is we have this little network of different groups that are all operating autonomously, but they're interlinking with each other, and there's nobody really controlling this. People are just doing what they think makes sense to do, and the thing is self-organizing. ChemRefer is another neat little outfit where they were actually reading our blog, and they will go and find articles relevant to research that you're doing. And so they actually found a paper on whether or not we could use phenols in our synthesis, and that's something that they initiated, they found, and actually contributed something positive to what we were doing. Okay, the other thing that's happening with this, because we have the research open completely, we can start to do collaborations with other teachers. Here's a collaboration with Beth Ritter-Guth, the High Carbon Community College, and she has English students who are looking at what we're doing in malaria and trying to make the connections from the humanities side. And their blogs are also public. So, there's a lot of leverage that you can have if you can get out of your WebCT and make your stuff public. I also have students in my organic chemistry class do extra credit assignments where they have to pick something we're doing that my grad students are doing in the lab and connect it to what we're doing in class during that term. So again, it continuously reinforces the importance of real work. Okay, now let me show you something really recent that I think you might find interesting. I haven't given up using blogs. All right, it's just that the blog is not necessarily the best place to store experiments, sequential experiments. What I use my main blog for is as an integrative tool where I will piece together what's happening on the wiki, what's happening in literature, and talk about milestones in our research group. Here's one recently where we have this compound. It's an isonitrile where we showed that it decomposes very quickly in the solvent that we're supposed to do the reaction in. And that's very troubling because other people have already used that compound and gotten good yields out of it. So the question is, how do you resolve that problem? Well, we are actually storing not just the NMR spectra. The spectra are what tell you, what the molecule is, but we actually have it in an interactive Java format. So we use this program called JSpecView that was written by Robert Lancashire. And what it does is basically allows you to expand any area of the spectrum. If you're a chemist, you know exactly what I'm talking about here. There's a lot of information in these spectra, and normally, if you just get a printout, you can't see the fine detail. And it turns out that the fine detail is where a lot of the interesting stuff is happening. So we have these available, and we are now actually exporting them in a CML format so that machines could read them as well. So a machine could go through and figure out what are the kinetics of this peak appearing and disappearing, and start to do automated discovery that way. So that's a direction that we are just starting to take now. All right, so I've shown you what we're doing. Let me show you some examples of what some other people have been doing in different scientific fields. In terms of raw experimental data, there is neurodatabase.org. I haven't found a corresponding blog where they necessarily translate the research, but they actually upload in Excel format all of the raw data for these experiments. I think they're with monkeys, and if you're in that field, it'll make sense. There is Org Prep Daily, which is a blog for organic chemists, and they will publish experiments that work and a few that don't. Here's an example of one where they were trying to make this compound and it didn't work, and it keeps going in the blog to explain what they actually got and why they think so. So that's a pretty good one. The other thing, there's a lot of discussion that's important for scientists that don't fit in the article format. One of them is vendor reliability. This guy was trying to repeat an experiment and noticed that depending on where they obtained the catalyst, they got different results. Normally, there's not a really simple way of doing that in the journal, and it's not an erratum. It's not something that's wrong. It's just a really useful thing to know. And so they're able to go and make a comment, and of course, anybody can, respond to that and say if they're seeing the same thing or not. So this is the kind of information that can be exchanged at least in organic chemistry. Now, there's another system you may have heard of, Open Wetware. It's a pretty large system that has different wikis for different research groups. One of the things that they have is protocols. Now, these are generalized protocols. So if you want to make, annealing primers, so you would actually go here and it would tell you in general this is what you do. So this is not the result of a definite experiment. It's just a general way to do things. They also have what they call lab notebooks. So far, what I've seen them use is information that's useful within their group, because the context of this page, for example, is not made explicit. So it would make sense to the group, what this corresponds to, what this means. But the information right now is not in a format where a stranger from elsewhere can just come in and start to contribute to it. At least I have not seen it yet.
I think that's intentional.
Is it intentional? Well, I think that they want to publish traditional papers, so I assume that it may be. It's also a pain to record everything, but since we actually use the wiki as the official notebook, it's not a duplication process. Students would have to do it anyway. Yeah.
Do you mean that you personally looking at that can't make sense of it because you're a chemist in biology, or do you think that because that makes perfect sense to me?
Well, from this experiment,
Sad as that may be.
Now, I mean, you can tell roughly what they're doing, but
I mean, I don't know what the plasmid is because that's not there.
right, exactly. What's the reason?
Right, so that's your point, is that, yeah.
If I wanted to contribute to this, what could I do? I can't look at the concentrations, you know. So the idea, basically, if you go on our Useful Chem wiki, is you can drill down. You have no our group has no special information. Everyone in the world has the same information that we have access to. We can drill down to any of the spectra, and you ought to be able to come to your own conclusions. Another thing is the discussing of hypotheses, and I think probably the best blog for this is our research. This is Rosie Redfield, and she goes into a lot of detail about very specific scientific hypotheses that she has with, I think, DNA uptake she's working on. And the thing about these kinds of blogs is that because they're so detailed, they're never going to be massively popular. And that's one thing I think that we have to, you know, learn to accept, that basically you can't have high quality research and a wide distribution. This is the classic law and tail. The more detailed, the more, you know, advanced the analysis, by definition, the fewer people in the world can actually understand it. So I think when we evaluate whether or not our blogs and wikis are working, we have to ask, are they reaching the people who are looking for it? And that may only be a handful of people, and that's okay, as long as they get it. Now, there's another part to this that I included because I think I wanted to come up with examples of people who are contributing real science, who are actually doing science using blogs and wikis. This is a blog by one of my collaborators, Aegon, and he basically publishes the code. You know, I showed you the stuff about the CML. So there are a few people who are actually writing code and, you know, coming up with the standards and all of that. And I think that's part of the science. They're not making chemicals, but they are enabling us to do what we need to do in terms of processing our chemical information. So there are a couple of good blogs. Rich Apodaca has a really good one. And again, I define good by actually putting code that you can use immediately. Christoph Steinbeck and Peter Murray Rust is probably the most experienced of these people. He's been working in the CML area for a long time. Like he built it. I guess what I'd like to end with this is that this is coming mainstream, which is really exciting. Chemical and Engineering News, which is the trade journal for chemists, this in the last summer had an article on open source science. This is actually my students here. And I think that really is important with respect to legitimizing the activity, trying to convince people who are not chemists that what we're doing, even though it is on blogs and wikis, it's not private diaries, it's actually, you can do real science with it. And I'm hoping that, you know, by coming here and talking to you guys, that we can, you know, forge even more collaborations between different groups. And that's it.